This video will discuss about gel electrophoresis. What is gel electrophoresis? Which is a method of separating large molecules like a segment of DNA. So it is a method of separating large molecules like segment of DNA. It is similar to TLC, but the principles involved are little bit different. So let's get a closer look at this technique. Mixture of DNA fragment that we want to separate and visualize. So here mixture of DNA fragment that we want separate and visualize. Gel electrophoresis enable us to do this, and its work as follows. This is the tray. There sits a slab of either polyacrylamide gel or agarose gel, which is immersed in an aqueous buffer solution. At one end of the gel, there is a series of well. This one is the well, and a number of samples can be loaded into this well, each of which is a mixture of some DNA molecules of varying length. So this one is mixture of DNA molecules of varying length. This apparatus is also equipped with electrode. Here two type of electrode is presented at each end, with the cathode or negatively charged electrode and the end where the well sit. Under that anode, this one is the anode or positively charged electrode at the other end. Once everything is loaded and ready to go, the current is here, power supply. Current is turned on. The DNA molecules are negatively charged. So this means that negatively charged cathode will repel the DNA molecules and they will begin to travel along the gel towards the positively charged anode, to which they attract. So this positively charged molecule towards travel positively charged anode. The things is the gel is sticky porous substance and the molecules have to migrate through the pores. To move along the gel is a process called sheaving. The larger DNA molecules, the more difficulty it will have to negatively through the force, which means the smaller DNA molecules will travel greater distance through the gel, while larger one will travel shorter distance in the same time interval. So smaller molecule DNA molecule travel greater distance. Larger molecule will travel shorter distance as the same interval. This process is so reliable and qualitable that we plotted that appropriate number in base pair in DNA molecules as a function of that precise distance is its travel during gel electrophoresis. Once separation is complete, the current is turned off and a DNA binding dye is added to that system that glows a fluorescent pink in UV light. This is how the data is gathered, which will show up as then band that sometimes resemble a ladder. If many different DNA molecules were present in the sample and each band containing thousands of identical DNA molecules of particular length. Remember, the further away from the well a band show up, the shorter DNA molecule is that has produced that band. This technique used to assess product of gene amplification as PCR. It can also used to isolate a specific DNA molecules of a mixture of sequence or further characterization via the technique called Southern plotting technique. In addition to separate mixture of DNA according to the length, and this technique can also be used to separate mixture of protein according to electrical charge, which offer information about the identity of that side chain or number of other way useful application. The simplicity and immense utility of gel electrophoresis make it a very important of any molecular biology laboratory today. So in this video we discussed gel electrophoresis. This one is the statement format. Thanks for watching.